But anyway, so two types of cells for each of the antibodies. Oops, but they differ. So CTLA-4 hits this effector CD4 cell that expresses ICOS. CD8 hits these T-bets that are effector cells, are actively proliferating, actively killing cells. PD-1 hits those effector cells and these exhausted cells as well, which fits with all the dogma, that you would hear about. So how do these interact? Because now almost everybody's getting both antibodies, right? Nobody's getting monotherapy with these antibodies anymore. So, what happens when you give both of them? Is it A plus B equals AB? You just see the sum of the two, and that's what we thought at first. These have slightly different things. You put them together, now you get the CD4, but with CTLA-4, which you didn't get with PD-1, and now you get these exhausted cells going with PD-1, which you didn't get with CTLA-4. So maybe it's that diversity that gives you, the superior, therapeutic effect of the combination. Well, no, that's not what happens, actually. What happens is very strange. So this is the experiment, same thing. We just added the fourth group here of the two antibodies in combination. And just to cut to the chase, these are these permanently exhausted, PD-1 high, LAG-3 high, TIM-3 high, really 3 strong. And you can see the data that another new experiment just showing the same thing. They're not changed by CTLA-4. They are changed by PD-1. But when you give them both, they go way down. You don't get more of them when you put the two antibodies together. You get less of them, okay, which is a little strange. But what about the effector cells? So this shows the effector cells, and you can see on the right, those are those differentiated cells that are perfectly healthy. You see a lot more of them, right? So than you do of the exhausted cells. So the question is, are we converting these exhausted cells here on the right to those in the left to those in the right? John Wherry would say no, but again, because of these epigenetic changes that have happened, unless you reverse that, they cannot go back. And so I don't really think it's that. I think what's going on here is that with CTLA, I'll come to CD4s in a minute, but those cells, the terminally exhausted cells, probably can't be turned around. But when you have PD-1 taking off the inhibitory effects that are on the terminally differentiated cells, and you're providing more easy co-stimulation at the front, it may be in the combination CTLA-4 by taking them both off, and particularly when they become functional with CTLA-4 around, they become, or when they have to go back and get co-stimulated again to avoid becoming exhausted, CTLA-4 blockade allows that. And so we may be creating a situation where the cells don't become exhausted anymore. We completely changed the biology. Now, we don't know that for sure, but I can tell you these data are absolutely solid. We've done this experiment, I don't know, many times. But in any event, and this is looking at those exhausted cells too. That shows you untreated or the monotherapy, the correlation of the increased numbers of those with small tumor size. When you give the combination, they're irrelevant. Not only do they decrease in number, but the ones that are there don't do anything anymore. So, again, we completely changed the way that it works. What about the CD4s? Well, they go up. I mean, they do have PD-1 on them. So when you block PD-1, they can go up in the presence of CTLA-4 at least, some more. And so, anyway, that's kind of the story. And I think that now, the part of the story, we need to start taking into account these differences in mechanism when we think about combining these two drugs and others. This is sort of a paradigm, perhaps. It says that, when you're putting things that together that affect different activation of T cells at different steps or inhibition of T cells at different steps, you could have different things happening. So anyway, this just sort of summarizes then what I've told you. It's not a combination that makes it more powerful. It's because of getting rid of those exhausted cells. So I'm going to close and just tell you about something. So these ICOS positive CD4s, these are, if you ask an immunologist, what's an ICOS? We've got a CD4 cell here. What it's ICOS positive, what is it? Well, they'll usually tell you it's a follicular T helper cells. These are cells that never leave the lymph nodes of the spleen. They're in areas where T cells and B cells are interacting to provide help to B cells so they can class switch immunoglobulins. They make type 2 cytokines, which wouldn't do you any good in a tumor. Or people say they're regulatory T cells. There's a subset of FOXP3 positive suppressor cells that have ICOS because it regulates IL-10. And so that's those. But Pam Sharma actually, in her very first pre-surgical trial in bladder cancer, showed that you get this huge increase in ICOS positive CD4s after you give N-acetyl-A4, but not after you give PD-1. And so, just to summarize this, so it went up in tumor and in blood. If you sort them, all of the tumor-specific cells that are making cytokines in the CD4s have ICOS. They're all that type. They're none of the conventional ones. They're associated with longer survival. We looked at about 150 of Jed Walchuk's patients that on the phase 3, and people that did not show an increase in those cells that was stable for the entire period of therapy had median survivals of 8 months, whereas the ones that did get them had median survivals of in excess of 24 months. So it doesn't guarantee to tell you who's going to respond, but it can tell you who's not going to respond. If you don't get those cells, you don't respond. In any event, put this into mice, did CTLA-4 therapy in mice who lack the gene for either ICOS or ICOS ligand, and CTLA-4 loses about 75% of its activity in those. So it really needs that PD-1. And if you mutate this tyrosine residue in a PI3 kinase binding motif, you lose the effect of increasing T-bet selection expression, and also you lose most of the therapeutic effect. So feeding into the AKT pathway through PI3 kinase, is required for this. And so this gave us the idea that maybe we can, by giving an agonist signal through ICOS, make CTLA-4 therapy work better. So here we went from fundamental discoveries, by looking, again, carefully at biopsies. In this case, it was whole tumors actually. This was done before they would let us do biopsies for research reasons. But anyway, so we asked whether you could enhance it. So this is an experiment that we did where we took tumor cells, in this case the B16 melanoma, and transduced them with with ICOS ligand or not, and then gave mice the wild type tumor, and then one or the other, either the vanilla vaccine without ICOS ligand or ICOS ligand, and then N-acetyl-A4. And these are the results, and you can see here very readily all the monotherapies are crunched together at the left, lower left. The red dashed line is N-acetyl-A4 plus radiated tumor cell vaccine that doesn't have ICOS ligand. The blue dashed line, the only difference is it has ICOS ligand. So by giving a signal through ICOS with this agonist, you increase the efficacy by about 4-fold. And if you do that in ICOS knockout mice, you completely lose the ability to do that. So it is, anyway, it's not its presence of ICOS, its ability to signal that does this. And we think because it raises T-bet and makes these cells into super killers. Anyway, we illustrate this anyway because we thought this was a good idea, got a patent, formed a company, and they've made it agonistic antibody, which is now in trials, and we'll see with that CTLA-4. We'll see what happens. It's too early. In any event, so now what we're going to see in the field is every combination you can think of. People ask me all the time, is chemotherapy gone? My answer is no. Where it really works, that's a good way to, kill some tumor cells, prime, you use them as vaccines, though, not as curative. Same thing for radiation. One of the trouble with both chemo and radiation is if you use enough to kill that every last tumor cell, you run into problems. And you don't need to do that if you just dust it enough to prime an immune response. In any event, there's also oncolytic viruses that we've had a lot of trouble. Cryoablation of a local tumor followed by CTLA-4 in mice will give you systemic immunity, where they'll reject every tumor that's in them. Anyway, so I think there's a lot of stuff going on, and we were talking how the next 20 years is going to be interesting. I think it's going to be really interesting for following this, but I will just stress again, is we can't afford to just be doing this stuff randomly. We got to have good basic science behind what we do. We don't have enough patience to do all the trials that would be necessary. In any event, I'll just close with this. This is what we've been used to in cancer therapy for the last 20 years or so. You get 1,000 people, you treat them, and then you look back statistically and see if you got a p-value, and if you do, declare victory. And there's nothing wrong with that if you could buy people some time. But we know from IPI, you could do that, but you could also have a tail on the curve of about a little over 20% who are apparently cured. And so the goal now, and this is aspirational, not data, but should be to move that tail up as high as we could get it. And I think there's a lot of optimism. I think that we know the basic rules. We're learning more every day by intensive investigation of patients who are getting different drugs. And I think that we're going to, like I said, with melanoma, we're at 60%. I wouldn't be surprised if we could get that up to 80, 90% in the next couple of years. And then with kidney, it's about 40% or so now. I think that same thing. There's new combinations that are being done, and they're quite promising. So anyway, with that, I'll close and hope people got a sense of optimism, and also more for companies to do to help us get this stuff done in a way that'll get these to patients faster.